Swayam Prabha. Digital India. Educated India.
Hello everyone. Welcome to the next lecture of second unit of plant biotechnology course. In this unit, we are learning about the haploid production through anther culture or the microspore culture, chromosomal doubling techniques, gynogenic haploids, chromosomal elimination techniques, and then the polyploids. My name is Manoj Sharma and I am working as an assistant professor of plant biology at Jawaharlal Nehru University, New Delhi. In JNU, I teach plant biotechnology course and genetic engineering at School of Biotechnology. These lectures have been reviewed by Professor Kashmir Singh, who is working as a professor of plant biology at Department of Biotechnology, Punjab University, Chandigarh. And this project has been sponsored by DTH Swayamprabha, MHRD, New Delhi. So in this lecture, I will discuss about the polyploidy and the production of polyploids. Then I will discuss about the classification of the polyploids, various mechanisms how the polyploids originate, and finally I will talk about the applications of polyploid in agriculture, plant breeding, horticulture, and other industries. So ploidy refers to the variations in the number of copies of the entire set of chromosomes, or it may be the variations in the individual chromosome numbers in the body cells of an individual. The complete chromosome set is a characteristic of any particular species and it is designated by letter X. And the phenomena or the condition of possessing three or more than three complete sets of chromosomes in the body cells is called as polyploidy, and the plants having such ploidy level are called as polyploids. Now if the number of chromosome sets in the cells is 3, these are called as triploids, and if the number of chromosome sets would be 4, that is X is equal to 4, they are called as tetraploid. And similarly, the number of chromosome sets may vary 5, 6, 8, and respectively they will be called as pentaploids or hexaploids or octaploids and so on. According to an estimate, somewhere around 50 to 70% of the angiosperms have undergone polyploidy during the course of the evolutionary process. According to some other estimates, this number may be even higher, as we have not accessed all the or tested the ploidy of all the angiospermic plants. In general, the polyploidy percentage is even higher in case of the pteridophyte plants. Polyploidy is common in plants, and in fact it has been a major driving force for the speciation of the land plants and also helped in the adaptation of these plants, angiospermic plants, to the dry conditions on the land. Now, before going ahead in the discussion of polyploidy or polyploids, let's learn about few terms which are associated with the ploidy. First is the diploids. So, if a cell contains two copies of chromosomes, that is the two sets of the chromosomes, it is called as diploids. Like here, we have two copies of each chromosome. So there are 5 chromosomes in total having 2 copies of each chromosome. The 2 copies of each chromosome, they are identical, they are same, and they are called as the homologous chromosomes. So we can say that there are 5 pair of homologous chromosomes in this cell, and this condition is called as a diploidy. And similarly, the plants having this ploidy level would be called as diploids. So in other words, there are 2 copies of the entire chromosome sets. This is the complete one set of the chromosome, and this is the second set of the chromosome. And they are each set, one set is designated as a with the term X. Next term is the monoploid. Now, if the cell contains only one complete set of the chromosomes, it is called as monoploid. So in this case, there are 5 chromosomes in the basic set of the chromosome, and therefore the monoploid number of the chromosomes would be 5 in this situation. The basic set of chromosomes in a species is designated by X, and it is a characteristic of that particular species. And the condition having a one set of the chromosome is called as monoploid, and the diploid means 2 sets of chromosomes, and triploid would have the 3 basic sets of the chromosome, and so on. Next important term is the haploid. Now, haploidy refers to the number of chromosomes present in the gametes of an individual, like egg or the sperm cells or the pollen cells in the plants. The chromosome number in the gametes is exactly half of the number of chromosomes in the somatic cells or the body cells of the individual, and it is usually designated by N. So N is the number of chromosomes in the gametes of an individual, and number of chromosomes in the somatic cell is double the number of chromosomes to the gametes, and therefore the number of chromosomes in the somatic cell is designated by 2n, and chromosomes in the gametes, they are designated by n. Like in rice, the 2n is equal to 24. That means the total number of chromosomes in the somatic cells of the rice, or each cell of the rice, like in leaves, stems, roots, it is the 24. Whereas in gametes, when the gamete will be formed through the meiosis, this number becomes half, so n will be 12 in case of the rice. Similarly, in the wheat, where the 2n is 42, and so n would be 21 in case of the wheat. Now, sometimes we confuse with the haploid and monoploid numbers. So it's very important to clearly differentiate and learn what exactly the difference between the monoploid and haploid. So we define monoploidy as the number of chromosomes in the basic chromosome set, or a single copy of the complete set of chromosomes in an individual. And haploidy is the number of chromosomes in the gametes of an individual. So let's let's study some examples and learn the monoploidy versus haploidy. So here is a diploid condition. There are 5 pairs of the chromosomes, that is 2n is equal to 10, and these are homologous chromosomes. Now, what will be the monoploid number in this situation? So if we distribute the chromosomes in the chromosome sets, there are 2 chromosome sets, and each chromosome set has 5 chromosomes. So the monoploid number will be number of chromosomes in a single set, and that is 5. How about the haploid number of chromosomes in this case? So in the gametes, the number will be half the number of the chromosomes of the somatic cells. So here gametes would carry 5 chromosomes too, as the total number of chromosomes in the body cells or the somatic cells is 10, and so n is equal to 5. So in this case, we can conclude that in a diploid individual, monoploid and haploid numbers are same, or we can say n is equal to x is equal to 5 in this case. So for the diploids, n is equal to x. They would have the same number of chromosomes. But the question is, does n always same as that of the x? So let's study an tetraploid individual. Now here is a tetraploid individual having 20 chromosomes. That is, each of the chromosome has 4 copies, and so 4 homologous chromosomes are there, and that's why it is called as a tetraploid individual. So what about the monoploid number of chromosomes? So the tetraploid individuals have 4 sets of basic chromosomes. So the monoploid number is equal to number of chromosomes in a single set, and again it would be 5. That is, it is designated by x. So x in this case, in this tetraploid individuals, where there are total 5 chromosomes and 4 duplicates of each, the x is equal to 5. So the monoploid number is same for the diploid or the tetraploid individual as the number of chromosomes in the basic set of the chromosome is same, and that is the 5. Now, how about the haploid state in this tetraploid individual? So by definition, haploids would have, or the gametes would have, half the number of chromosomes of the number of chromosomes in the somatic cells. Here in the somatic cells, we have total 20 chromosomes, that is 2n is equal to 20, and so gamete should have 10. So n should be 10. So in this individual, though the x is equal to 5, that is the number of chromosome in a single set, that is 5. However, in the gametes, that is the n would be 10 chromosomes, and so basically monoploid and haploid number is same for the diploid species, but as the ploidy changes, monoploid and haploid number also differs at a higher ploidy levels. Next is another term is the monohaploids versus dihaploids. Now these terms are associated with the number of complete basic chromosome sets in the gametes of an individuals usually, whereas monoploid is also kind of a haploid individuals. So in case of the diploid plants, where there are 2 sets of chromosomes, its gametes would have only 1 set of chromosomes, that is half the total number of chromosomes. And if the gametes have a single set of chromosomes, these are called as monohaploids. So this gamete is also called as monohaploid as it has a single set of chromosome. However, in case of the polyploid individuals, the situation will change, it will be different. Like here is a tetraploid individual that has 4 sets of the chromosome. Now in the gametes, when the meiosis will occur, it would result in the half the number of chromosomes in the gamete. So the gamete would have 2 sets of the chromosome. Therefore, n is equal to 2 sets or the 2x. We can also say n is equal to 2x. And this condition is called as dihaploid. So now this term is different than the double haploids. Dihaploids and double haploids are different. Double haploids we studied earlier when we were talking about the haploid production. So there we were culturing the haploids, and then we induced the doubling of the chromosomes to generate the condition of diploidy. And so the double haploid was the term associated with the sporophyte that was generated from the haploid plants after doubling the chromosome number. And their gametes would have only one set of chromosome, whereas dihaploid is the term associated with the gametes of the tetraploids, where there are two sets of chromosomes, and similarly there will be different sets of chromosomes as the ploidy level will change. So now next is the how we classify or categorize the ploids. So based upon the composition, ploidy can be categorized into two groups, that is euploids or aneuploids. So first is the euploids. Now if the cells have chromosomes in exact multiples of the basic chromosome number, these are called as euploids. So we can say that euploid genome complements have multiples of x, like it would have 2 copies 2x or 4 copies 4x. So these are called as these are categorized as euploids, and if there are 2 copies we usually call them as diploids. However, if the number would be higher, 3, 4, 5 sets, they are usually called as polyploids. So we have diploids which contain 2 copies of basic chromosome number. So it may have configuration like AA, that is the 2 sets, or BB or CC, where each letter represent the chromosome set, where A, B, C represent the different species, and so on. Now next is the polyploidy. So the condition of possessing 3 or more than 3 complete sets of chromosome in the body cells is called as polyploidy. So there may have 3 copies, that is the 3x, and it represent the triploidy. Similarly, there may be the 4 sets in the tetraploids, 5 sets in the pentaploids, 6 sets in the hexaploid, and so on. Depending upon the type of genome copies present in the nucleus, these are further categorized into 2 groups, that is autopolyploids or allopolyploids. So the first is the autopolyploids. Polyploids that carry multiple copies of the one complete chromosome set of a species are called as autopolyploids or autoploids, and the phenomena is called as a autoploidy or autopolyploidy. That means all the copies of the genome are similar to each other, like AAA configuration. That means there are 3 copies of the A genome and it is a triploid. And similarly, in case of the plant having the ploidy level BBPP, each cell have 4 identical copies of the genome of the species B. Here is the parent karyotype of the microspore and megaspore mother cells, which undergo meiosis and form male and female gametes respectively. And here the 2n number is total is 8 chromosome in a particular cell, and upon meiosis the gamete should have 4 chromosomes, that is n is equal to 4. So after meiosis gamete will be formed where number of chromosomes will be 4. However, due to the meiotic error, reduction in the chromosome may not occur during the gamete formation, and so it would result in the gametes that would have same number of chromosomes as that of its parent. When these unreduced gametes they fertilize together, it results in the doubling of the chromosome number. Here the chromosome of the same individual are duplicated and the tetraploid individual is created. And this kind of hybridization where the chromosome sets of the same species are duplicated, these individuals are called as autaploids. Now, depending upon the number of the genome copies that are present, these are named accordingly. Like here in this case, plants have 4 copies of the genome and hence it is called as autotetraploid. If the unreduced gametes will fuse with the normal gamete, so normal gamete would have n is equal to half the chromosome, that is 4, and unreduced would have 2n, so the resultant will be the resultant individual would have 3n, that is a autotriploid. Natural autaploids have been identified for several plant species of commercial importance like potatoes, peanuts, coffee, and banana, etc. Similarly, spontaneous autaploids are very useful in the horticulture industry too, and have been identified for several plant species that are used in horticulture like tulips, gladiolus, or dahlia. And this is usually because of the Giga's effect, which is very common in polyploids. So basically the phenomena of the enlargement of the body organs, principally because of the accumulation of the moisture content, is known as Giga's effect. So in the horticulture industry, large flowers are desirable, and therefore polyploids having the large organs or the large flower are preferred and used at a large scale. Among various types of natural autaploids, tetraploids are more common in nature, and several are used as a commercial cultivar in agriculture as well as the horticulture industry, or even several of them they are used for the production of metabolites in other industries. Second category is allopolyploids. So the polyploids having multiples of more than one complete basic chromosome sets are known as allopolyploids. Like the plants with the ploidy level AABB is a tetraploid as it has 4 sets of the chromosomes. Here each letter represents a copy of the chromosomes. Earlier we talked about the autotetraploids where we discussed this configuration AAAA, where all the copies of the genome originated from the same species by the duplication of the chromosomes, and all the homologous chromosomes are identical. However, in case of the allotetraploids, these two have 4 copies of the genome, but these originated from more than one species. Like here in case of the tetraploid of AA and BB configuration, different colors are used to differentiate the chromosomes contributed by different species. So these chromosomes have been contributed by species A, that they are represented by A, and these dark colored chromosomes, they are contributed by the species B. Therefore, they originate by combining of the entire basic chromosome sets from more than one species. Similarly, we have AA, BB, CC configuration or the ploidy level. It's a hexaploid as there are 6 sets of the chromosome, but here 3 different genomes are there. So it means that in addition to A and B genome from 2 species, C genome is contributed by a third species or another cultivar. And this phenomena of combining genomes from different species during hybridization is called as the alloploidy, and the plants having such configuration are called as the allopolyploids. So here are the parent cells of species A and species B that are 2n, and the 2n number is equal to 8. These will undergo meiosis and will produce haploid gametes where n would be equal to 4. These haploid gametes from different species may fertilize to form a zygote with 8 chromosomes. Though the zygote contain 8 chromosomes like its parent cells, but these are not the homologous chromosomes as these are contributed by 2 different species, and therefore these zygotes having chromosomes from 2 different species would be genetically unstable. Therefore, for the stability or the genetic valence, an immediate chromosome doubling would be required. So it would result in the doubling of the number of chromosomes, and as it carry the genomes of 2 species, these are called as a allotetraploid. Natural alloploids do exist in nature. In fact, several alloploids are used commercially in the food industry like wheat and oats. In the fruit industry too, like the strawberries or blueberries are the allopolyploids, and several other industrial crops are also used for the various industrial purposes which are allopolyploids like tobacco, sugarcane and cotton, etc. Now let's have a quick look on the origin of the hexaploid wheat. It is very well studied how it has been evolved. So wheat is an allohexaploid, and its genome comprises of basic genome sets from 3 different species. It was originated from diploid ancestors, and therefore 3 diploid ancestors have contributed their genome copies, that is A, B and D genome sets, for the generation of modern hexaploid wheat. It has generated through the 2 hybridization events that occurred during the different time points in the past. First hybridization took place between Triticum monococcum and Triticum sessi. Both of them were diploid, having 2n is equal to 14, that is total of 14 chromosomes were there in their somatic cells. Genome of Triticum monococcum is A type, and genome of Triticum sessi is designated as a B type. So Triticum monococcum contributed the A copy of the genome, and sessi contributed the B copy of the genome. Gametes from these 2 diploid ancestors had 1 copy of the genome set, and they fused together to form the zygote having 14 chromosomes, but again these were not homologous, but they belonged to 2 different species. Therefore, this hybridization between the gametes was immediately followed by chromosome doubling that resulted in the AABB ploidy level and created a tetraploid species of wheat. So it had 4 complete sets of the chromosomes. However, these sets are contributed by two different species. So it is called as allotetraploid with 2n is equal to 28. So total 28 chromosomes were there, and it is the species is Triticum turgidum. So it is also called as durum wheat and is valued for its high gluten content and is grown for industrial purposes. It is specifically used for the preparation of pasta and bread flour. And this particular hybridization event, which led to the creation of this tetraploid or allotetraploid wheat, that is Triticum turgidum, it happened somewhere between 10,000 to 30,000 years ago. Now, somewhere around 8 to 10,000 years ago, another hybridization event occurred between the tetraploid turgidum species and another diploid wheat species, possibly Triticum toshii. Its genome is designated as a D genome. So that is for the Triticum toshii. So the gametes of the Triticum turgidum would have A and B copy of the genome. That is because it is a tetraploid, so its gamete would have two sets of chromosomes. And when it fused with the gamete from the Triticum toshii having the D genome, it created a hybrid with the three genome copies, that is the A, B, and D. And again, the chromosomes of these genome sets were not homologous, and therefore, for the stability or the genetic balance, doubling of the chromosome was essential. So for this hybridization between these dihaploid gametes and the haploid gametes, it was immediately followed by the chromosomal doubling events that resulted in the hexaploid modern wheat, where total number of chromosomes are 42, that is 2n is equal to 42. And this event took place somewhere around 8 to 10,000 years ago. So basically, for the creation of the modern hexaploid wheat, three diploid ancestors have contributed their genome sets. That is, A genome is contributed by the Triticum monococcum, Triticum sessi contributed the B genome, and then the D genome was contributed by or donated by Triticum toshii. In the current situation, cultivated wheat has hexaploid as well as tetraploid and diploid varieties. And Triticum acetybum is the modern cultivated hexaploid species. Triticum turgidum is the cultivated tetraploid species, and it has two subspecies, that is these are which are cultivated. These are durum wheat and emmer wheat, and they are specifically grown for their gluten content and used for various purposes in the food industry. And finally is the diploid encorn wheat, that is the Triticum monococcum. It is the most ancient cultivated wheat variety. As per some estimates, it was cultivated as early as somewhere around 35 to 40,000 years ago. So that's how the evolution of hexaploid wheat occurred from their diploid ancestors. Next is the origin of polyploids. So what are the various mechanisms of the origin of polyploids? Like we just studied the hexaploid wheat, how they were created. So what could be the different kind of mechanisms that result in the polyploidy? So the polyploidy may result from the non-reductional meiosis during the gametogenesis. That is, it produces the unreduced gametes. That means this division is not reductional, and the number of chromosomes in these unreduced gametes will be similar to the number of chromosomes in the parent's somatic cell. Here is our parent microspore or megaspore mother cell, which undergo meiosis and form haploid gametes. So here is a 2n parent cell, and in the gametes, ploidy will be n, that is the half the number of chromosomes in the somatic cell. Now, under certain circumstances, specifically the environmental stress conditions, this reduction do not occur, and the ploidy of the gametes would be the same as that of the parent. That is 2n number of the chromosomes. Now, when these unreduced gametes fuse together, that is 2n plus 2n, or they may fuse, that is the unreduced gametes, 2n gametes may fuse with the normal gametes having a half the number of the chromosomes as in the normal condition it occurs. It results in the individuals with the higher ploidy level. When the unreduced gametes fuse together, they generate the tetraploid individuals, wherever when the unreduced gamete will fuse with the normal gamete, it will create a triploid individual. Next, another mechanism of polyploid creation is the polyspermy. So it is also called as the polyspermaty, and it represents the condition when more than one male nuclei or the sperm nuclei or the pollen nuclei, they fuse to the egg cell. So it creates polyploid condition, and a repeated polyspermy can create the plants with the higher ploidy too. Now, polyploids may arise from the chromosome doubling. The doubling of the chromosomes can occur immediately after the fusion of the gametes, and this results in the complete polyploid individual. We just studied the evolution of the wheat, where when the gametes of the two different species they fuse together, immediate doubling of the chromosome is required for their functionality, as otherwise these are not homologous or genetically they will be imbalanced. However, the chromosome doubling may occur locally too, like in the somatic cells, and hence also called as the somatic doubling. Now, if the somatic doubling occurs during the zygote, it results into the complete polyploid individuals. However, if the somatic doubling occurs in the somatic cells, like if it occurs in the meristematic cells or the apical meristem cells, it may result in the polyploid chimeras. If it occurs in maybe some cells from which are no more used in any growth, like leaf cells or the stem cells, it may not just result into any kind of major developments or the major polyploid organs. And if the somatic doubling occurs in particular cells that later develop into the flower bearing branches, then it may be passed, this polyploidy may be passed to the next generation easily, as the seeds developed from these polyploid flowers would be polyploid too. So the somatic polyploidization, because of the endoreoduplication, is common in plants. Endoreoduplication is a condition where during the cell division, though the DNA synthesis takes place, but it is not followed by the cytokinesis. So this means though the DNA doubles in the nucleus, it is not followed by the cell division, and so it may lead to the ploidy level or the higher ploidy level inside the cells. However, it depends upon if the new plants are regenerated from these cells, if having the polyploidy or not. Next is the characteristics of the polyploids. That is the morphological or the phenotypic characters of the polyploids that can be a characteristic features for the identification of these plants. So the polyploid plants have usually large cells as compared to their diploid progenitors. This enlargement is specifically visible in the meristematic cells. Then their leaves, specifically the leaves of the autopolyploids, they are thicker, broader, and shorter. However, the other organs and sometimes the leaves too, they increase in size due to the Giga's effect, that is the enlargement due to the accumulation of moisture content. Autoploids usually resemble to their parents morphologically, whereas the alloploids tend to exhibit intermediate phenotypes between the two parent species from where they originate. Then the metabolism of the polyploid plants is usually slower, and also their growth rates are retarded as compared to their diploid progenitors. Slow growth rate is usually attributed to the lower concentration of the plant hormones in these polyploid plants, and specifically the concentrations of the auxins. Slower metabolism and lower growth rates help these plants to perform better or grow better on the degraded soils or the soils which are nutrient poor. Polyploids tends to flower late in the life cycle. Their flowers are usually bigger and they stay for longer period of time as compared to the diploid progenitors, and all these characteristics are very much desirable in the flower industry for the economical benefits. Ploidy also affect the chemical composition of the plants, and usually these plants accumulate higher amount of the secondary metabolites. Even the artificial polyploidy has been induced successfully to increase the commercially important secondary metabolites like the artemisinin, which is an anti-malarial metabolite, and its accumulation increased several folds when the tetraploidy was induced. Similarly, the significant increase in the nicotine content, that is up to about 20% increase in the nicotine content, was observed when the tetraploidy was introduced in the tobacco plants. Next is the applications of polyploids. Now, polyploidy has been a major force or the driving force in the course of the evolution and speciation in land plants. It is estimated that somewhere around 50 to 70% of the angiosperms they are polyploids, or even some authors suggest that this number may be even higher than the 70%. Generally, polyploids are more tolerant to the environmental stress conditions, more specifically like the drought or the cold conditions, as well as the higher resistance to the pathogens or the pests. In the characteristics, we discussed that the polyploids usually has higher accumulation of secondary metabolites. So usually this enhanced production of the secondary metabolite is associated with the resistance to the biotic pathogens. Polyploids have been used to produce the superior crops. Specifically the triploid crops, they are more popular. Like several triploid fruits are available in the market, like seedless bananas or seedless watermelon, and we will talk about more detail in about the triploids in our next lecture. Further, the development of the sterile cultivars has a huge market in the ornamental industry. Sterile ornamental plants usually stay for the longer period of times, and therefore they are desirable for the landscaping purposes, and also these do not become invasive as these are sterile and do not make the seeds. Further, their flowers are larger and they also stay for the longer period of times, and so they are commercially valuable in the flower industry or the horticulture. Production of large fruit has huge applications in the fruit industry. Organ enlargement is a common feature of the polyploidy, and hence most of the polyploids bear large fruits. For example, the triploid apples. Now, induction of the triploidy here has increased the size of apples significantly without compromising the quality of the apples with respect to either taste or the nutrition. Chromosome doubling help in overcoming the interploidy variants. It plays very important role in the evolution of allopolyploids. These various to the hybridization due to the differences in the ploidy level can be simply removed by chromosomal doubling, and these events have occurred naturally as well as they can be induced artificially for the breeding purposes. It is well known that the polyploidy enhances the production of secondary metabolites that make these plants robust. Large number of metabolites are used in various industries like the pharmaceuticals, cosmetics, and the food industry, and so on. And so the polyploidy used to enhance the production of metabolites of the commercial interest too, like narcotic metabolites in the case of cannabis, datura, or the Atreupa plant species. Artemisinin, which is an anti-malarial metabolite. So its production was increased several folds when the tetraploidy was introduced. Similarly, the enhancement of the production of corticosteroids has been achieved in the Dioscorea by introducing the ploidy or the higher ploidy, and similarly terpenes production has increased in the Ocimum and Mentha because of the change in the ploidy level or higher ploidy levels. Overall, polyploidy is an exciting phenomenon with a lots of practical potential. Many of the crops are polyploids, and polyploidy is playing a very important role in the breeding programs for enhancing the heterozygosity, development of the robust cultivar with respect to the tolerance to the environmental stress conditions or biotic stress conditions, and so on. So, in summary, we talked about the polyploids and several genetic terms or the ploidy related terms associated with the polyploids. We discussed about the classification, various groups of the polyploids. Then we also talked about the various possible mechanisms through which the polyploids can originate, and finally we talked about the various potential applications of the polyploids. In the next lecture, we will talk about triploids in a little more detail. Thank you.